Good morning everyone. Let's come to another session with Michelson and Stahl's experiment. So this is one of the experiment which one of the most scintillating experiment ever to describe what is the nature of the DNA, how it is conserved, okay, and whether it is highly how it is being, which of the strand you have, the DNA has double strand, so which of the strands are being transcribed, whether it's a single strand or double strand and so on. So the learning outcomes is you will come to know about the applied experimental results of Michelson and Stahl's experiment, okay, and how each strand of a DNA molecule serves as a template for the synthesis of new or complementary strand. So how my DNA replicate? There had been so many theories, and how it is being replicated. One is a conservative nature, which is old, of course, the DNA old thinking, like the double strand goes to one daughter strand intact and other daughter cell get a new copy. So again there was something called dispersive nature, where parental strands are dispersed into two new double helices, right? Again they are being dispersed, and both daughter cells would receive old and another one new, but would involve in cleavage of the paternal strand, most complicated and least likely. You cannot find, it does not happens, in such a way. So another method which was semi-conservative nature, which says that each daughter cell receives one new strand or one old strand, one new strand and another is the old strand. So each strand serves as a template to synthesize a complementary strand. So how is it possible? See, you see, conservative nature is completely one of the DNA will go into the daughter cells. Okay, dispersive nature, half of them will go one point is there where you will have a one strand, one fragment of the strand, another fragment of the strand, and so on. So it's highly dispersive. But you see, these are most likely unlikely. Okay, but here you could see it's being semi-conservative. It means one of the strand is going into the daughter cells and it's synthesizing a new strands. So to prove it can be semi-conservative, and which is much more likely, there should be some experiment. So Michelson and Stahl came up with an experiment which is called the grew E. coli, because E. coli is a genetic material. So they grew E. coli in medium that contained a ammonium chloride with N15 labeled, right, and another strand was not N15 labeled, okay, and they grew for many generations, okay, within the, so that all the bacteria were radioactive, and the natural form of which N14 was much lighter than N15. So if the DNA is being separated, okay, by the weight centrifugation, you would also find, okay, the DNA which is having N15 will have it will sediment at the lower phases, okay, and one which is lighter will sediment at the higher phase. That's logical to understand. So DNA made with N15 is heavier than with the N14, and will form a discrete band. You can find a discrete band. So that is what he did. So he grew a E. coli and he had to extract all the DNA from the cells, and he had centrifuged. Okay, now you are finding all the N14, right? Now again he has taken only the N15. Now he has taken N14. Now you are radio labeling it. So when he is radio labeling it, all the bacteria is radio labeled with N15. You are finding all the DNA which has been centrifuged, you are finding N15, okay. But now you see N15 cells and transfer to N14 media. Now that's what they have made, a combination of N14 and N15. Now what he found is he extracted the DNA. Now he was saying there was some double. You see in this you are finding N14, and here you are finding N15, which is a lower sedimentation, but here you have N14 and N15, which is somewhere lying between N14 and N15. So this is how it was proving. Now not only that, again he replicated the cells of second time to produce a second generation of daughter cells. This is the first generation. Now what he found, he was finding all the bands together. One was N15 and another was N14, and also he was finding N14 and N15, right? So this is how he tried to prove that DNA is not dispersive or conservative, but it's semi-conservative in nature. So this is what he was using, N15 and N14 experiments, right? So you see density, the sedimentation density. You have N15 which will have a density here, and N14 which will have density above. So the simple what's called generally you are using the blending or blenders experiment and so on. So you'll find the gravity and so on in this you'll find. Okay. So this is how it proves see you have a DNA and one of the strand is old strand and the new strand is being synthesized here on opposite to this. It is old strand and a new strand. So this is what's called semi-conservative nature. So remember that the replication is bidirectional and semi-discontinuous. So in prokaryotes, bacteria and most of it's bidirectional and so on. So here all these are important, to understand how it is conserved and so on. Okay. Hopefully you understood the concept of semi-conservative nature of nucleic acids, especially the DNA. Thank you so much.